Hello and welcome to our presentation on a hydrogel injectable scaffold for periodontal gum restoration and reattachment. My name is Erin and my partners are Sammy, Alona, and Elizabeth. So the problem we chose to focus on is the issues surrounding periodontal gum and the progression of tooth decay and loss over time. Gum disease is very common in people over 65, with 20% with untreated tooth decay and another 68% with gum disease. This is likely to progress to affect the tooth as seen in the graphic below. There is a large need for a proactive and preventative measure that facilitates gum restoration and reattachment to promote loose tooth fixation and decrease overall recession ahead of time. A device that facilitates this growth would promote and strengthen the longevity of the patient's health. Currently, dental implants and surgeries are very costly and cumbersome as well. So gingivitis is a predecessor for periodontitis. Gingivitis shows itself as a red, swollen, and inflamed gum from plaque and compiling, beginning in the gingival sulcus, which is a neck of soft tissue that connects the teeth. The sulcus typically ranges from 0 to 3 millimeters, and they are healthy pink in color. Once plaque and biofilm begins to form in the sulcus and mineralize, the gum has a response that becomes swollen and red, forming gingivitis. This process is reversible, but quickly leads to periodontitis if left untreated. Periodontitis is where the attachment is lost when the sulcus depth increases, eventually leading to tooth-supporting alveolar bone being lost. This can lead to tooth loss, an unappealing look, and bite collapse with other systematic health problems. So the motivating factors of our device was to develop a method and procedure that naturally improves the attachment of the gum and fibers, which would increase the fixation and restoration of the tooth. Generate the growth of the gum to minimize the receding gum line with a uniform integration to improve the health of the mouth. The implant additionally should be less invasive than grafting and offer a long-term solution that's better than the ones that are currently on the market. By combining the knowledge of tissue integration, attachment, facilitation, and mechanical properties, we developed our product.
All right, so moving on to current treatments, starting with dental implants. These are made out of 3 parts. Firstly, the prosthetic, which is the tooth-colored, typically made out of ceramic piece that is seen by everyone else. Then fixated to the jawbone is the implant itself, and those 2 pieces are connected by something called the abutment. This is a really cosmetically appealing approach. However, it is non-absorbable and it doesn't actually fix the mechanism of periodontal diseases and is also known to cause irritation in the gums. Next, we have gum grafting. There are 3 types of this, starting with gingival grafts. These are smaller pieces of gum that are excised and then transplanted to somewhere else in the mouth. Next, then we have connective grafts, which are deeper grafts, and these are more used for deeper volume restoration. And then lastly, we have pedicle grafts, which are cut lines in the gum, and then a flap is folded downwards. All 3 of these methods of gum grafting are used to slow down progression of periodontal diseases, but they are more of a cosmetic approach and they don't actually help fix tooth fixation, but it does use the patient's own tissue. Then lastly, we have an area to which literature has pointed towards recently, which is enamel matrix derivatives. These are injectable matrix proteins, typically made from either bovine or porcine models, and they're often used in conjunction with other techniques as it's not a standalone technique yet. And it provides the ability for cells to hopefully ingrow. But as I said, it is not a standalone technique.
The way we plan to approach the problem of periodontitis is via utilizing the techniques pioneered via the Chao Pinhole method, combined with an injectable bioinductive scaffold composed of stem cells as well as other additives to facilitate reattachment of tooth to the bone and to the gum. The Chao Pinhole method was pioneered by Dr. Chao, and it is a far less invasive method to combat periodontitis or gum reconstruction surgeries. As you can see, the incision that is made is very small, and a tool is used to loosen the gum of the diseased tooth. By loosening the gum, this gives room for the doctor to inject the collagen strips that will then dissolve and grow into gum. What our group plans to do is, instead of injecting collagen strips, we will inject our bioinductive scaffold that will not only create gum growth, but also facilitate tooth to bone reattachment. The instrument we plan to use is a tilted syringe that will be 27 gauge, will be at a 45-degree angle and composed of stainless steel. This instrument will be injected into the gum, into the root of the diseased tooth, where we will inject our bioinductive scaffold. Our scaffold is mainly composed of a chitosan hydrogel. Chitosan is a carbohydrate that is said to be biocompatible, biodegradable, and have low cytotoxicity, and is therefore preferred for injected scaffolds. It is also shown to have antimicrobial properties due to the interactions between the positively charged chitosan and the negatively charged bacterial cell wall. Our scaffold will also be composed of collagen, specifically type 1 collagen, which supports cell adhesion and is often used in connective tissue as it plays an important role in tissue to bone interaction. In order to supplement the collagen, we will add fibroblast growth factor, which upregulates collagen and increases the stability of collagen RNA. The stem cells we plan to use are dental pump stem cells, which are stem cells often used in dental procedures, which we will harvest from the patient in the root area of the tooth. They will then proliferate and further facilitate tooth to gum reattachment. We will also be adding concentrated growth factor. This is a novel growth factor combined of platelets with a fibrin matrix, as well as growth factors and leukocytes. It has shown to be effective in regeneration by facilitating the degradation of alpha granules in platelets. Concentrated growth factor has also shown to have a positive effect on osteoregeneration. We hope that by combining stem cells with a collagen-based scaffold with growth facilitating proteins, we will expect tooth to gum regeneration as well as the growth of new periodontal tissue. The way we plan to fabricate the chitosan hydrogel is through the combination of chitosan, acetic acid, and deionized water, incubated for 24 hours in a 4-degree temperature in order to facilitate anti-gassing, then injected into a cylindrical mold which will be frozen, then immersed into a first gelling solution composed of NaCl and TTP, then immersed into a second gelling solution composed of NaOH. We will then wash the hydrogel and it will be ready for use. In order to introduce the dental pulp stem cells into our scaffold, we will extract the dental pulp from the patient or use previously extracted dental pulp if the patient had done so. The dental pulp will be centrifuged and cultured. After the cells are cultured, they will be placed into the injectable scaffold.
Model, which is the rat. This will just give us the basic inflammatory response in regards to our scaffold. The next animal model we plan to use is canines. Canines are very often seen in literature to be used for testing for periodontal treatments. Particularly, they have been used to test various growth factors and extracellular matrix proteins, similar to the ones that we plan to use in our scaffold. So this is a very applicable animal model to our project. And then finally, we plan to use a large animal model. This animal model will be non-human primates. Primates are very good for animal models because the anatomy in a primate is very similar to humans. And these primates have also been seen in periodontal treatment research particularly treatments that include different biomaterials. So all of these animal models are very applicable to our project, and we think that they'll give us a lot of information on how our scaffold works. So overall, the benefits and limitations to our project, there are many benefits, including the surgical technique in itself. Because it is a minimally invasive technique, this will be a very comfortable procedure for our patients, and they'll have minimal pain and a really quick recovery. Our scaffold itself is beneficial and unique because it's a bioinductive scaffold. And we hope that this scaffold will promote gum tissue regeneration as well as improved gum to tooth attachment. So this scaffold will improve and better the function of our tissue. And we expect that this scaffold will be able to withstand molar restoration up to 1,000 newtons. In terms of limitations of our project, one of them is the high cost, which I'll touch up on in the next slide. Another is the dental pulp stem cell sourcing. This can be done in various ways, and our preferred method is not for everyone, and it can be expensive as well. And we do recognize that most people with poor oral hygiene also have financial troubles. So this is something that we plan to look into further. And the last limitation of our project is the duration of the surgery, and how long these effects will last after the surgery is done. And this we plan to mitigate through testing. So for the cost of this scaffold, like I said, it is pricey. The surgery itself is around $3,000. And then the contents of our scaffold, as well as some other lab equipment, vary. This makes our total for the whole surgery around $7,000 to $8,500 per patient. And while this is pricey, we do think that this bioinductive technique will outweigh the cost and will be more beneficial to the patient.
All right, so moving on to sterility, this has proven to be an incredibly large challenge for hydrogels, as they're obviously incredibly sensitive to changes in temperature. So a major concern would be denaturing the ECM proteins or disrupting the 3D scaffold itself. But with that, a lot of literature has pointed towards the use of supercritical CO2 and said that it is an incredibly promising technique to be used with the sterilization of hydrogels. And we hope to use that in conjunction with sterile processing conditions. And then the group also hopes to provide a characterization of all the different sterilization methods and their effects with the hydrogels to fully understand their effects on the hydrogel. So moving on to competition, starting with an implant. The first one is one of the leaders on the market. It's a titanium zirconium alloy, and it uses acid etching to improve hydrophilicity. It's used with a variety of abutments and prosthetics, but as I said, it is one of the leaders on the market. However, one of its limitations is it does not actually fix the periodontal disease at all, and it just replaces the tooth. Moving on to the Straumann membrane flex. This is an occlusive membrane. It typically is used in gum grafting procedures, and it really helps strengthen the graft site and helps with the wound healing in that area. Again, it does not help with actually gum restoration or tooth fixation, but it does help with the patient experience. And then lastly, we have Emdogain, which is the leading injectable matrix protein. It's a pre-filled syringe. It's sourced from young porcine tooth germs, and it's typically used in flap procedures. So this is a really invasive approach. However, the Emdogain product, it does really help with the wound healing and is known to really speed up that process. But once again, it's not actually helping the mechanism of the injury and the periodontal ligaments decay. So we're going to open it up to questions. Thank you everyone for listening. And then here are our references.